Okay, so today is our last lecture, and what I thought I'd do is give you a practice quiz, which will be worth no points, but it'll give you an idea whether or not the amount of studying that you are doing is enough. And I want you to take this quiz kind of seriously. I don't want you to work or talk about the questions with the people that are sitting next to you, or I want you to see how you would do if the quiz was for real. And some of you will say this quiz is a piece of cake. Some of you will say, I've never had questions like this before. So these will be real world questions that you'll get on the first exam. And I get that some of you have never had test questions like this. And it's going to take you maybe a test or two to kind of get into the rhythm or the routine. So I'm just going to ask you to prepare just like the way I told you to prepare, which means using your lecture outlines, combining information that we've talked about, things that we've drawn, things that we've watched. When you make this one gigantic study guide, then you should be good. And notice that I never said the big green book at all. You could use the big green book to go back and say, you know, I didn't understand when Mike was talking about homeostasis. Let's go see how the big green book explains it, and use that as a resource guide. But you don't have to study the green book at all. You just use it as a resource. All right, so let's try this as a practice. So if you have a blank piece of paper, let's try this. Again, do this individually. Give you a couple minutes per. So read it and then write down what you think the answer is. So don't get thrown off by the number 13. That was question 13 on an exam a few years ago. Okay, everybody good? Can I move to the next one? Okay, here we go, the next question. Question kind of makes me hungry a little bit. Okay, everybody good? Question number 3. This is a good one. Okay, everybody good? This one is dedicated to my wife who gets poison ivy just by looking at it. Two of the above. I hate those.
I did.
Okay, everybody good?
No.
All right. All right. Oh, now this is my kind of question. It says, read the following statements. How many of these statements are false, you'll have one or two of these on the test. So all you have to write down for this practice test question is a number. A little hard taking these kind of test questions when you can't write stuff down. On the exam, you'll be able to write all over the test and work things out. It's kind of hard to do that sometimes when it's on the big screen. Okay, so I think that's it. So let's go through them and see how you did. So this is as real world as it's going to get. So you have an idea now of what to expect. Since you're walking down the tough streets, your muscles keep a form of energy stored, so that means humans store energy. What macromolecule stores energy in the muscles? Well, you could say a carbohydrate does. That's a macromolecule. But it's definitely not glucose. Glucose is a monomer, right? It's a building block. Galactose, I don't even think we talked about that, so if we never talked about it. I wouldn't expect you to magically know that, so it can't be that one. Starch, well, we talked about that, but we said that's the storage fuel tank for plants. And we said glycogen is the storage fuel tank for people, so the answer is D. You got that one right, you should be very happy. Lard is an animal fat. It says the key word here, it's solid at room temperature, which means you should stop and say, okay, that means it's a saturated fat. So it's not unsaturated, it's not polyunsaturated, it's not unsaturated, it's definitely not a steroid. So the answer would be C. I mean B. Sorry. The answer is B, saturated fats. Saturated fats are solid at room temperature. Unsaturated fats, the good kind, are liquid at room temperature. 2 for 2. How many of you are 2 for 2? About 20%. Oh, this is one that required lots of thinking. No, it didn't. No, it didn't. It doesn't matter what I said the person is doing in a wheelchair, upside down, bedridden. It's always dealing with anatomical position. So the nose is always superior to the mouth. So my wife gets poison ivy by looking at it, by picking up my clothes, by bumping into our dogs. And she'll scratch it, and it spreads like wildfire. The more she scratches, the more it spreads. The more, which is a positive feedback. See, I can bathe in it. I don't get it. All right, last one. Here we go. How many are false? Let's go through them. Carbs are hydrophilic. True or false? In harmony, everybody. True. You only knew that because that's in your table, right? As the number one characteristic that you wrote down. And if you're saying what or what, that means you're going to have an awful day next week on Tuesday. All proteins are enzymes. True or false? False. That's true. You are correct. That is false.
It's false.
Oh, wait a minute. All proteins are, no, there are proteins that do other things that are enzymes. Lipids are fearful of water. True. Remember, oil and water don't mix. Lipids, you can also remember, are non-polar. All enzymes are proteins. That's true. 100%. Nucleotides are made up of nucleic acids. It's just the opposite. And you would only know that answer is if you were studying your table. Nucleic acids are the polymer macromolecule. Nucleotides are the monomer. Butter, which is solid, is an unsaturated fat. False. We just had a question on that one. Amino acids are bound together by a very weak bond called a hydrogen bond. True or false?
False.
They are held together by peptide bonds. Substrates have active sites. True or false?
True.
False. Proteins have active sites for substrates. Remember, the substrate is the key. The active site is the lock. So it looks like we have a little work to do. So if you got all of these right, then you should say, I am doing what it takes. If you got less than half of these right, you'll probably do a 50% or worse on the test on Tuesday, unless you start changing your strategies. You know, there's probably 5 or 6 people who come to office hours every Tuesday and Thursday. Some of you are just asking questions, some of you are just coming to listen. I mean, both of those are great. But if I never see you in office hours, I'm assuming that you're mastering it. Like, you have no questions. So if you don't show up to office hours and you get below a 50, that's a big mystery to me. That should never happen. Yes? Oh yeah, great, thanks for reminding me. So on Tuesday of next week is our first exam. So remember what I said, I will start the exam at 5 o'clock. You don't have to be here at 5. The exam is intended to go for an hour and 20 minutes. So you get more than that naturally. If you're a slow test taker, you like to triple check, you're always the last one to turn in an exam, that's you, then maybe you should get here at 5 so you have no stress about time. No stress about time. And yes, Mary.
People have lab after the test.
Of course. That's
Figured.
awful too.
Figured.
It's awful. So after you burn out your brain on the test, you gotta go upstairs for a 2-hour lab. That stinks. Any other questions about the exam? Oh, here's another one. The exam, the multiple choice is a Scantron form. Listen, so you have to bring a pencil. You have to bring a pencil. Everybody hear that? Bring a pencil. You can write all over the test and write cheat sheets on your test and make little drawings to help you remember things. Usually when people do poorly on the first test, I try to ask them politely to come see me, and I'll get out the exam and we'll go over some of the questions that you missed. And usually, listen, this might be a new thing for some of you, is that usually people who do really bad are people who don't write on the test at all. So if there's a good test question, it should be a question that you say, I could narrow it down to 2 choices. Like, 3 of them are weird. I don't even know what you're talking about. Cross those out. But 2, I've narrowed it down to 2. And if I look at your exam and I see you crossing out the weird ones and you've narrowed it down to 2 and you choose the wrong one, I'm gonna say, you know what, you're close. You're gonna do better. But if you're picking the crazy ones or you're just guessing, I don't know. So prepare, study, study, study. All right, let's get into today's last topic. So on Tuesday, we talked about cells, we talked about transport, and we said that when we look at a cell, there's 3 major components, and we spent most of lecture on Tuesday talking about the plasma membrane and transport across the plasma membrane. We spent the last 5 minutes of class saying the word cytoplasm, which was the liquidy environment that contains all of the organelles. And just as a reminder, you're responsible only for the function of the organelles that you had to learn in lab. So if you had lab on Tuesday, you say, I know what you're talking about. If you have lab tonight, you'll get a good idea of which ones you have to know. So we're going to say that the cytoplasm was Tuesday, the plasma membrane was Tuesday, but the nucleus, the third one, is tonight. So that's where we're going to start. We're going to start by saying that the nucleus in many high schools is usually called the brain of the cell. And I don't think that's a good description. I want you to think of the nucleus as more like the architect of the cell. So it stores information, it sends out specific instructions, or you could say it creates a blueprint on how to build things. That's a good one. So that's what the nucleus does. So we're going to say that its main job is to store and to communicate. There we go, store and communicate. Up on the big screen, you can see something called chromatin. And chromatin is the stuff inside of the nucleus that usually picks up the stain. Remember methylene blue? For those of you who had lab on Tuesday, it makes the nucleus of your cheek cells look dark. So this chromatin is a combination of DNA, RNA, and protein. DNA, RNA, and protein. And you can see that when we think of some of the functions of DNA and RNA, it's to hold, carry genetic information. In fact, when you look at chromatin, you can see that chromatin can condense from the DNA molecule. We'll talk about DNA in a minute. Some of you recognize this from maybe high school as being the double helix or a twisted ladder. But you can see that it kind of coils up and twists, and this is what chromatin looks like. But the final product is that it starts to condense into structures called chromosomes. So a lot of times maybe in high school, people drew chromosomes like this, and that's a little wrong. When you think of a chromosome, I want you to think of chromatin, look, twisting and wrapping itself into one very long structure. And then you can say, well, there's regions on this chromosome, look, that maybe code for certain traits. We'll talk all about this next semester, but we call these regions genes. You may have heard of that. And this is what a chromosome looks like, really. But one of the challenges today that we're going to talk about is mitosis, something we briefly introduced in lab on Tuesday. And during mitosis, the cell replicates, it divides to make another exact copy. So if we make a copy of a cell, at some point we have to replicate the DNA and the chromosomes. So before cells, look at this, before cells start to go through mitosis, they go through a step where they replicate the chromosome. So you have an exact copy stuck together. And it does kind of look like an X. So in high school, when you drew an X, you really drew a chromosome that was getting ready to divide. It made a copy of itself. So this structure, a chromosome, is what we find housed inside of the nucleus. And the nucleus is a structure that has a membrane. And I would write down in your notes that it's very similar to the plasma membrane in that it is selectively permeable. Or when we talked about the plasma membrane, I think I even said selectively leaky. Some things can go in and out, but not everything. So let's take a look now a little more closely at this twisted ladder, this DNA molecule. And one of the things that you can see is that it is a large macromolecule. It's a nucleic acid. And it's made up of smaller subunits called nucleotides. So here's what I want you to be able to do. If I would ask you on the test on Tuesday, which I'm going to, so here's another example where I'm giving you a free question. And I would say, what are the ingredients of a nucleotide? All you have to do is tell me it is a sugar, it is a phosphate, and it is a base. Those are the three ingredients. Do you have to draw them? No, no way. This is not biochemistry. If I give you a picture of a nucleotide like this, could you identify which one is which? And you should, ready? Because when we say phosphate, it has a P. When we say sugar, it looks kind of like a house. And then this structure up here is what really is different from nucleotide to nucleotide. And we call this the base. So here's the phosphate, sugar, and base. There's a chance that I might put a diagram of a nucleotide, and you have to label which one's the sugar, which one's the base, and which one is the phosphate. And you would never have to tell me what the base is. You know, don't say, oh my gosh, I have to remember what adenine looks like. So, a nucleotide, the monomer of nucleic acids. Okay, so far so good. Let's look at all of these these nitrogenous bases, one ingredient of a nucleotide. And I would never ask you to identify these by themselves. But usually when we get into the bases, we start using letters. And so sometimes we refer to the bases, these, as the language. In fact, what I've written up here is the DNA language. So there's a base that we're just going to call A. It stands for adenine, thymine, cytosine, and guanine. You don't need to know those official names. You just need to know the letters. So it's these letters that attach to a sugar and a phosphate to make a nucleotide. So far, so good. So the RNA language is a little bit different. We have A's, and we have T's, and we have C's and G's, but there's something that's unusual. This T gets replaced with a U. So there are no T's in RNA language. Okay, so now you know the language of both DNA and RNA. It's 4 letters. Yeah?
Are we gonna have to be able to recognize them by the individual?
No, you never have to recognize them, no. You just have to know what the language is. Now what's interesting about these letters, let's focus on the DNA language, is that when you build a DNA twisted ladder, that means you have 2 rungs, 2 sides. And what keeps both sides of the ladder hooked together? It's hydrogen bonds. And notice that the hydrogen bonds attach, look, these bases. And the bases don't line up randomly. So let's write this down, that when we talk about DNA language, we're gonna say that A's always hook up with T's, and vice versa. And C's always hook up with G's. Always. In fact, let's do this. Between an A and a T, there's 2 hydrogen bonds. But between a C and a G, look at this, there are 3 hydrogen bonds. In RNA language, we have the same kind of what we call complementary base pairing, which means they're not random. So instead of saying a T, we have to say a U. Remember, there is no T. So U's hook up with A's. C's, just like in DNA language, hook up with each other. Okay, now, when we look at this business,
Do they bond the same way with RNA?
that's a good question.
U?
That's a great question. So I drew their complementary base pair, but one of the big differences between DNA and RNA is that RNA is single-stranded, which means it's smaller. So the only reason why I drew their partners is because when DNA communicates, you have to know what kind of is the appropriate response. So DNA is double-stranded, which means larger. Okay, so far so good. Let's take a look at this double helix. So the circle over here represents a phosphate. The house represents, which one? Sugar. And then we have a base. Phosphate, sugar, base. Phosphate, sugar, base. Okay. The phosphates, this is kind of interesting. The phosphates are very acidic. And that's why we describe this molecule as an acid, right? DNA, deoxyribonucleic acid. It's all about the phosphates. Okay, so far so good. We know a little bit about DNA. We're not going to go crazy. So as I said before, DNA function is to store stuff. It's also to communicate. So the way we communicate usually is that we tell the cell to make something. In most cases, what we're making is a bunch of proteins. We drew over here by me that in terms of DNA, it can store genetic information in the form of traits, hair color, eye color, whatever. And all together, when we look at the information stored in DNA, we call it the genome. And you may have heard of the Human Genome Project, or you may have heard of small animals like mice having their entire genome being recognized and identified. Those are big, big, exciting things in science. So what we have to do now is talk about the communication component in a little bit more detail. In order to do that, we have to appreciate, again, some of the differences. RNA is smaller than DNA. RNA exists in a number of different forms. We're going to call it transfer RNA, messenger RNA. Oh, there's even more than that, but for this class, just know those two. We said RNA is smaller because it's only one single-stranded chain. It's not a double helix. We said U's replace T's. So far, so good. And when we talk about the main function of RNA, it's to help interpret the DNA message. All right. Now we get to do some drawing. Some of my favorite things. I would say that this is gonna take up 3/4 of the page of paper. So let's allocate 3/4 of the page. So I'm gonna draw over here kind of an overview. I'm gonna draw an overview of what we're gonna be talking about for the next 10 minutes. So we're gonna talk about the process called transcription and translation. These processes usually cause a lot of sweating in students. I can remember learning about this in high school and just saying, I have no idea what they're talking about. This is way over my head. But we're gonna do it in a slow way, and it's gonna make it really understandable, I hope. And I think one of the best ways to learn this is to maybe redraw it. So maybe that's a good way for you to learn. So we're gonna start with this cell, and we're gonna say that DNA, which is in the nucleus, wants to communicate to the cell to make some more proteins. And when I say that, most of you should think, okay, really the cell is trying to make some more enzymes. Remember, proteins can be a lot of things, but enzymes like their number one function. Unfortunately, remember what we said about the nuclear membrane, it is selectively permeable. It can allow some things, but not everything. And since DNA is a double helix, 2 strands, it's too big to get out of the nucleus. So how does the nucleus send a blueprint to make more enzymes? It has to change, ready, into RNA, which is only 1 strand, and RNA is small enough, look at this, to get through into the cytoplasm. Now that right there, you have to say, oh, that's pretty cool. So to change DNA language into RNA language, this is called, here we go, transcription. What I would write next to the word transcription in your notes is that it takes place in the nucleus, and the purpose is to change DNA language into RNA language. Okay, if you get that, that's really important. I didn't remember that in high school. No way. It was so complicated. That's not complicated. That makes sense. Okay, so now we have RNA language out into the cytoplasm, but RNA is not an enzyme, so we have to change RNA into protein. So in order to do that, we have to undergo, look at this, translation, which takes place in the cytoplasm. And the purpose is to change RNA language into protein language. All right, jackpot. So far, so easy, I hope. So now what we have to do is talk about some details and how that really works. This might get a little crazy. So the way I always teach it is that I try to blow up the nucleus. So I'm going to draw the nucleus. Here's half of the nucleus. And then here is the plasma membrane. Does everybody get that? So this is the nucleus. And this is the cytoplasm. Okay, so here's what I'm going to do on the test. I'm going to give you some DNA language, and I'm going to ask you to take it through the process of transcription and translation. And I think there's 6 questions in a row on the test dealing with this drawing. So drawing may help, but make sure you understand it, okay? So I'm going to make up a piece of DNA language. I'm making this up, so don't say where did this come from. Okay. So go ahead and write that piece of DNA language in your nucleus, that exact fragment. And so what I want you to do is, since this is DNA, I want you to create the double helix for me. And I want you to write the complementary base pairing for that single strand of DNA. So go ahead and do that right now. Fill it in. Taj, come on. Don't look at me like I'm speaking Chinese. So what goes with an A? You put that on the other side. Don't tell me. Do it. Come on, Rox. So do it. And then crank it up a notch and say, which ones have double bonds and which ones have triple bonds? It's right there. Should erase it. So Taj, ready? Hit me with your best shot. What is the other strand? Taj, loud. T, A, G, C, C, G, T. 8 plus, Taj. So this right here will probably be question number 53 on the test. Whatever. Question number 37. I don't know. So that was easy. But now we have a challenge of saying this piece of information, just this piece, is going to code for a protein. But it's too big. So we have to change the DNA language into RNA language. So here's how it works. I want you to just take this brand new fragment that we created, and I want you to write the RNA equivalent underneath it. So do that.
And that happens in the nucleus too?
Yeah, I'm not outside the nucleus yet. Dominic, I'm going to ask you for this answer, so hurry up. Okay, so Dominic, oh, you know what? I'm going to put it in a different color. Dominic, go. A,
U,
U,
C,
C,
G,
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 C, A, U, A.
100%. A plus, that's question number 28. And what we've just done is we have transcribed. So that process from here to here is transcription. Yay for that. Okay. So we have a fragment of RNA, and unfortunately, when we create a fragment of RNA, it is, like with our English papers, it is an unedited form of RNA. Omkar, do you have a question?
I was just wondering, how do you know which DNA strand gets?
We're always going to do the newest one. Yeah, good question. Okay, so now we have an RNA strand, but it is unedited. The first thing about this strand that we can do is describe it as messenger mRNA. This is what's going to leave the nucleus as the message. But it's unedited, so we call it, ready? We call it pre. I'm inspired. I feel like I should be Superman or something. Pre-mRNA, which means it's unedited. So let's be a good 12th grade senior student and edit this piece of RNA language. All we do is go like this, watch. We go like this. 1, 2, 3, 1, 2, 3, 1, 2, 3, So we organize our pre-mRNA, which is another way of saying unedited mRNA, into groups of 3. And we call these groups of 3, ready? Codons.
What if it doesn't have to?
Great question. So if we have a situation where we don't have 3, like this case, look, AU, it only has 2, we're going to edit this sucker out. So our final mRNA that's going to get ready to leave is, look, AUC, GGC, U. And this is what we call our mRNA. Take the pre off of it because we just edited it. Each one of these codons now can exit the nucleus. And so we call these codons that can exit, we call them exons. And underline the word or the letters EX because they can exit.
Do they exit altogether? Or
Well, yeah, that's a good question. They do exit as a strand.
Okay.
Yeah. The AU up here that gets stuck inside is called an intron because it stays in the nucleus. So that's some lingo. For some of you, it's the first time you've ever heard it, so let's review this again. So we have a single strand of DNA that we made into a double strand. We use that newly formed double-stranded piece to make an unedited form of RNA language. Dominic did it great. And we have to edit it. And the way we edit it is we put it into groups of 3, and we say that once we have groups of codons, they can exit. So we call them exons. So look what happens. This goes through the cytoplasm. So we have AUC, GGC, and AUU. So now we're right here, over here by my finger. Look. And so the next step is to translate. Notice the words transcribe and translate usually have to do with languages. Like you've heard of old transcriptions of older textbooks, or you say, hey, I learned a new language. Let me see if I can translate that letter that was written to me in Spanish or something. So you're changing the language. All right, so now we have another type of RNA, and we call this transfer RNA. And transfer, or tRNA, is what assembles what we call the opposite of these codons. So we're using RNA language, and we're going to say the opposite of this codon is a UAG and a CCG and a UAA. We're almost there. So the transfer RNAs assemble the opposite of these codons, and it makes sense then that we call these the anti-codons. They're the opposite.
So they, not transcribe,
They're translated.
so they translate.
Yeah, but we're not done yet. They're in the process of translation. Because these opposite codons, these anti-codons, then, here we go, code, each one codes for an amino acid. So once we have these anticodons, they are not then translated into amino acid language. And guess what we do? We stick them together by bonds only found in between amino acids, which were called what, everybody? Good, peptide bonds. And what level of organization do we have in terms of protein structure do we have now? Everybody know me? Single-stranded, so we just have one strand of amino acid. What level of organization is that called? Come on. Primary. Remember, primary is just amino acids. Secondary is they're folding. Tertiary is in the big blob like a popcorn kernel. So what we have done is we've taken DNA language, we've transcribed it into RNA language. Why? So it can get through the nucleus. And then once we're in the cytoplasm, we can change that RNA language into amino acid or protein. That's translation. Now in some schools, maybe in some high schools, instead of writing down amino acids, you would have to look up to see what each one of these anticodons, what specific amino acid does it create. We're not going to go there. This is not biochemistry, this is just AMP. So we're going to say that this is where we're going to stop. Okay? So here's your challenge. I give you challenges, hopefully you do these. Remember, the first challenge was to make a table, and I've checked about 20 of your tables. Yay for you. The other question that I told you to do is to think about, what would happen if a fish was placed in, salt water or something. This challenge that I want you to do this weekend, and I want you to send me a picture of your results. Text them, don't email me. Because I want you to make up your own strand. Just make up a strand. Do it like 3 or 4 times to practice. Then make the opposite DNA helix. Make your pre-mRNA. Make your final mRNA. Make your anticodons. And then that'll be it. That'll be it. You've just gone through 2 processes that I would say most students find really hard, but I hope you're not thinking that it's too hard. So practice this. Everybody good? Okay.
Could you go through, what did you say, the primary, secondary, and tertiary?
So remember when we talked about proteins as being a macromolecule? I said in your table you had to write something down about characteristics. And I said proteins can exist in 3 different shapes. The primary was just a bunch of amino acids in a big long chain. Then I said proteins can start bending and folding. And then Dominic, I said tertiary was something like this, a big blob. Yeah. And we talked about proteins being really specific in terms of temperature. And remember what we said? That if a protein was denatured because of temperature or pH, it could go all the way back down to its primary structure, which is what we just drew on the board. So by having a primary structure, it now allows it to build. Okay, so yay. I would say that is one of the toughest things from today's lecture. But it's not tough, and you're going to practice. So, oops, sorry. All right, let's see what I do. So take a look at this diagram. Don't take a picture of it, but you can see the whole process again, where DNA comes from, look, or codes for messenger RNA, which is unedited, and then we have messenger RNA squeezing through the nuclear membrane and then translated into protein. Okay, we're done. Transcription, translation, yay, yay, yay. So all of this up here is something we have talked about. I did not talk about the enzyme that is used to make RNA, but don't worry about RNA polymerase. We know what pre-mRNA is. We know what the introns. Remember, those are the ones that get stuck because they stay inside. And the exons are the ones that can exit the nucleus. Okay, so far so good. This is an actual picture of what the process looks like in the cytoplasm. And you can see that we have mRNA kind of floating around out here in the cytoplasm. And we have organelles called ribosomes, which you'll learn about in lab. And these are the structures that kind of are involved with forming that, look, three-dimensional glob. So this is like a strand, and now it's starting to form something implicit. Do not take a picture of this, but you can see that we have a double helix. Getting into look at this, we have a single, we'll look at one strand like we did in the board, making an RNA copy, which then codes for anticodons. And then you can see that each anticodon creates an individual amino acid, and putting all the amino acids together is the primary structure of protein. You do not have to know anything from this, so you can put an X to this in your lecture outline, but I like talking about it. This says that in order to make some proteins in unbelievably adverse conditions, whether it's maybe hot temperature or lots of acid, sometimes you need help with this process. And look what they call them, chaperone proteins, or proteins that can handle stress or heat. It's pretty sweet. So just so you know that sometimes this process needs a little help. I will not ask you anything from this slide on the exam. Okay, now we get to move into our last big topic, and that's DNA replication. And one of the things that you can see here is that we have a double helix that literally unzips. Back, I guess, 2 or 3 lectures ago, I said that when we look at G's and C's and A's and T's, we have hydrogen bonds, which we're going to say for the next test, the first test, is the weakest of all bonds, yet they're involved with one of the most important biological molecules in the world. And now you understand what the answer is to that question that I asked you to write down 2 lectures ago, is that the DNA double helix is constantly unzipping or breaking apart. So we don't want really strong bonds, right? We want hydrogen bonds that do the job but can break apart really easily. Write this word down. It's called helicase. Helicase. What a great name. This is an enzyme that unzips the DNA double helix. Or you can write down on the exam that helicase breaks the hydrogen bonds. Ooh. Question number 3 or 4 from just today's lecture that I'll give you on the exam. Helicase breaks the hydrogen bonds. And what's the purpose of breaking hydrogen bonds? Take a look. It creates 2 separate strands. And once we have 2 separate strands, we have this gigantic popcorn kernel called DNA polymer, right? We know what that is. And since it's an enzyme, we put ACE on it. DNA polymerase, DNA polymerase. And all it does, look, is it takes one side of the DNA backbone and it does just like Taj did. And add the complementary base pairs to make, look at this, a brand new double helix. Same thing with the other side. So when we're finished with helicase and DNA polymerase, we have two exact copies. Money, when we're trying to divide cells or to heal parts of our body. So I hope you get that. It's kind of cool. So DNA replication, sometimes it's called semi-conservative because it's using 1/2 of the DNA helix. Rick, I think you're right. I'm starting to warm up a little bit in here. You can see that the DNA helix is something that forms chromosomes. I drew an X over here. So whenever we think of DNA replication and the process of unzipping and making two different copies, it really brings us into our next topic, which is mitosis. So, I'm going to erase this. And I want to talk a little bit about mitosis. Mitosis is a process where one cell divides to make two, and this is important, put 3 stars by it, 2 identical cells. Yep, and if they're identical, sometimes we refer to that genetically as being daughter cells. It's kind of sexist. They're never called son cells, boy cells. They're always daughter cells.
Never seen a son give birth.
That's true. That's true. So mitosis, the process of making 2 genetically identical cells. I want you to write this phrase down. I pee on a mat. Oh, that sounds disgusting. But you'll never forget the stages of the life cycle of a cell. This represents the life cycle of the cell. Do me another favor and draw a dotted line separating the I from the P. The I stands for interphase. And this is the longest stage of the life cycle.
Is that really in mitosis? Oh, I thought the
Yeah, you're one step ahead of me.
Okay.
So interphase is the longest stage. It's where DNA replicates, which we just talked about. But it's not, and underline this and put a star by it, it's not a part of mitosis. It's kind of like what we would call pre-mitosis. I'm getting it. So, replication happens. Remember what that means? Helicase, DNA polymerase. Then it goes through some stages. Prophase, metaphase, anaphase, and telophase. And when telophase is finished, we have 2 cells. So starting back about right here, I would say somewhere between A and T, we have a process called cytokinesis, which means cell splitting. Now for lecture, that's probably good enough. Good enough. In lab, you have to recognize each one of these stages. So in lab on Tuesday, you were supposed to have a picture on your phone of each one of these stages. I don't know about this, listen to this, if you were in lab on Tuesday, did you take a picture of the models over there on the counter?
Yes,
Did you take a picture of the poster above the models?
yes.
Okay, then you'll be okay. So tonight, I'm gonna give the Thursday night group those same suggestions. The other thing you can see about interphase, in addition to it being the longest stage, is that you can actually divide interphase into 3 categories, and I just want you to write down one. It says that S stands for synthesis, and this is where the replication takes place. So if you want to get really specific, you tell me about interphase, you say it's pre-mitosis. You can also then jump ahead and say, ready, that DNA replication takes place during S phase of interphase. That's money right there. Okay, so you will see these different stages of mitosis and identify them in lab, and hopefully it makes more sense. For those of you who had lab tonight, then those 4 people who had it Tuesday. Tuesday I said mitosis, and some of you were like, what? But now this is a good review, hopefully. So here's some functions of mitosis, and put a star right each one of these. Definitely need to know this. So when we think of cell division, we think of embryonic development. As soon as the sperm hits the egg, it starts to undergo mitosis. When we think of puberty and growth, we say cells are undergoing mitosis. When we think about girls in the 6th grade going through puberty and all that estrogen, man, just think, most of the girls in 6th grade were way taller than the boys. Then we think about boys in like 8th grade, 9th grade going through puberty and all that testosterone. Oh, then they start growing. So in other words, estrogen and testosterone increase mitosis. I wish I would have you next semester, because when I talk about estrogen and reproduction, then it makes sense why when women use estrogen therapies, maybe after menopause, or I bet some of you are on some type of birth control right now, which is estrogen, there's an elevated risk, listen, that estrogen in your birth control pill or in your menopausal treatment could encourage some cells to go through mitosis again. This time form cancer, not gonna make you grow taller.
Whoa. So you said birth control could make someone cause cancer?
I'm saying that estrogen in birth control causes a elevated risk in some people. Yeah, 100%.
Why they don't state that? You know how they go
Oh,
there
they
and
do.
sign that?
They
It's
do.
on the
They
warning.
do.
State that.
Oh, they do.
They do.
They do.
I've never seen that. Is that
Yeah.
what-
So since we're on that tangent, there's different levels of birth control. For example, my daughter's on birth control right now to help with acne. And so that level of estrogen and progesterone would probably not be enough to help prevent pregnancy, but it's a lower level, so I'm hopeful that it's not a big deal. When my wife and I were using birth control back in the day, I was always anxious for her to get off of it, because not that she has cancer of that type that runs in her family, but it just puts you at an elevated, it might be a smaller elevated risk, but there's still a chance. So, in addition to tissue growth, it's good in terms of mitosis in that it repairs worn out, dead, or injured cells. So a lot of times people use the word mitosis in the same sentence as healing or replacing. So that means some cells do not heal or undergo mitosis. For example, brain tissue doesn't really do that. If someone has a stroke or someone has a spinal cord injury, yeah, we hear about some people recovering, but a lot of people don't because they're unable to go through mitosis. So put this in stars, not every cell can go through mitosis. Okay, so in lab, we went through the whole process of looking at the stages of mitosis. It involves looking at chromosomes. Take a look at this. Here you can see a chromosome being organized, and the chromosomes are lining up and then being pulled apart and separating into two genetically identical cells. So, one thing that I want you to think about is that first drawing that we did over here. When we drew a chromosome that looked like this, we're going to call this chromosome an unreplicated chromosome. So this is before it gets to S phase of interphase. But then once it goes through S, DNA replication, S for synthesis. And now we have an exact copy. So through mitosis, they pull apart. One goes this way, one goes this way. They form two cells that have exactly the same information. But there's some lingo that's associated with this chromosome that gets a little tricky, and I want to go over that. We're going to say that each chromosome consists, after it's been replicated, consists of two chromatids. So when I say chromatid, I want you to say two identical strands. If I drew the chromosome before S phase, then we would say this is one chromosome, but zero chromatids. So chromatids usually are held together. They're identical. And they're held together by kind of like this staple called a centromere. So if a centromere is present, think of it like a staple or a paper clip. They're holding two chromatids together. If there is no partner, then there's no chromatid. Which then leads to an interesting question. Look at this. If you look at the stages I, P on a mat, look what happens when we get into the end of this process. We say that the X's line up on the metaphase, but then they get pulled apart. So once they get pulled apart, there's no longer a pair, right? They're all separated. So after they pull apart, here in anaphase and telophase, there are no chromatids. They're only chromatids when they look like X's, like this. So sometimes students kind of struggle with that idea. Chromatids, maybe write that down. Chromatids only exist when they look like an X. When they get pulled apart and they no longer look like an X, they are considered chromosomes, their own chromosome, but no chromatid. There's a really cool research kind of idea. So cool that I think I wish I would have done this about 15 years ago, so you guys still have time to do this. Is that what we know about chromosomes is that on every chromosome, there is a cap present called a telomere. These are so cool. And what we know about telomeres is that when cells replicate, here we go, through mitosis, each time, look what happens to the telomere. They start to get slightly smaller. And when they no longer exist, or you can say when they get worn away, worn down, then the cell can no longer replicate. and they become something, and write this down, put a star by it, they become a senescent cell. Which means it's a cell that lives but doesn't replicate. It's a cell that just kind of exists. It kind of gets in the way. Yeah.
Can you please spell that? I can't see it.
Senescent. Senescent, yeah.
So just conceptually, is that aging? Like when
100%. So this is one of the ideas behind this research is that if you know that you have worn away telomeres and your cells are no longer able to replicate, then maybe that's a sign of getting older. Alexis, do you have a question?
Can you go back over and say what this?
I can't hear you.
The telomere you said.
So telomeres are on the ends of chromosomes, and they get shorter every time the chromosome replicates. And when they get all the way worn away, the cells cannot replicate anymore. So there's a company, ready for this, that says you can, in 2019, send off a blood sample or a saliva sample, and they will measure your length of your telomeres. And they will tell you how well you are aging. They're not going to tell you, listen, when you're going to die. They're going to say, how are you doing?
But can they predict when you're gonna die by?
They can predict that maybe you won't live as long. So listen to this. I would suggest doing it around age 30, personally. I'm 50, listen, I'm 51, so I'm past this point. But at age 30, you could have this test done, it's like $200. Listen, they'd send you back the information, and they would say, whoa, you're 30? You have the telomeres of a 15-year-old. You are living healthy, you've made, listen, wise decisions. Man, your telomeres, you have the potential to live a long time. Or maybe you say, you know what, I made some bad decisions in my life, but I'm changed. But your telomeres may show that. So you might say, hey, I'm only 30, my telomeres are shorter than what they should be, so it's going to be a wake-up call. You know, maybe it's a time for you to say, I'm going to cut back on whatever and whatever, and do more of this. So I think it's pretty cool. Rob?
I have 2 questions. So basically you said once our telomeres are done, we die?
No.
Oh.
I said once they become senescent cells.
Then we
Senescent
die.
cells say that the cells no longer function. So things start to slow down.
Okay, so they just slow down,
Yeah.
okay. And the second question, so the chromatides are only in DNA?
Chromatids.
Sorry, they're only in DNA and not RNA?
Right, they're only chromosomes.
Okay.
Mary, do you have a question?
Where do they take the sample from?
I think your mouth. I think it's a mouse. So we'll watch a few seconds of this video clip. It's pretty cool.
Are there any diseases like?
Oh, yeah.
Can you make the length of your telomeres longer?
Back now at 8:19 with more of our special series, How to Live to 100. This morning we're focusing on the signs of longevity. Reaching the century mark sounds tantalizing, but is it possible for everyone? Here's NBC's chief medical editor, Dr. Nancy Snyder. There are approximately 70,000 centenarians living in the United States today, and some scientists say that the first person who will live to see their 150th birthday has already been born. But who are these centenarians going to be, and can we increase our chances of becoming one of them?
The science of aging is becoming really popular because there's so much more out there as a consumer to take control of their health and manage their aging.
In 2009, 3 scientists won the Nobel Prize for their research on telomeres, perhaps one of the most exciting developments on the aging forefront. Telomeres are structures on the tips of chromosomes that shorten as people age. Think of them like the plastic tips on a shoestring. People with shorter telomeres are more likely to develop illnesses and die earlier. The science is far from complete, but some laboratories have seized on this opportunity and are offering tests that measure a person's telomere length. Spectra Cell Laboratories is the only lab in the United States to offer telomere testing. The cost? $290.
It can serve as a wake-up call for some people. It's a measure of how well you are aging on the inside. So for some people, it can tell them that maybe they're aging faster than they expect, and maybe they can make some major lifestyle changes.
Hi, Howard. I decided to be my own guinea pig and with a simple blood test, see what my telomeres had to say about me. I hope I slide out at home plate. That's all I'm asking for. But if our telomeres are short, is there a way to make them longer? That's where Dr. Gil Atzman and his team at the Aging Lab at Albert Einstein College of Medicine are focusing their research.
So that's an interesting question. If your telomeres are shorter, what can you do? And so that brings us to our last slide of the night. And the last slide deals with, or the last couple slides deal with, the idea of what do we do when we have cells that don't ever run out of their telomeres. They never stop growing. We call cells that don't stop undergoing mitosis, we call them cancer cells. So even though cancer is a horrible word, we've all heard it, it's actually a type of cell where the telomeres never get short. So maybe we can learn from a cancer cell how to apply that to our cells. Maybe the key to longevity and long life is to make sure that our telomeres stay long. Interesting. You can see two words up here that we'll talk about more, but when we talk about metastasizing or angiogenesis, those words are unbelievably scary. It's saying that the cancer cells have divided and moved to a brand new location. And if a tumor cell, remember what a tumor is, it's just an overabundance of mitosis. If the tumor cell starts to grow its own blood supply, then that's even scarier. You never want to hear, oh my gosh, look at this tumor, it has its own blood supply. So when we think of cancer, now we know. We can learn from it. Don't forget that. Good essay question. We can say that cancer cell is nothing but out of control mitosis. So yeah, you can see that there's a lot of things that cause it. Okay, so that will bring us to the end of the information for this exam. Remember again, if you came in late, that I'm starting the exam at 5 o'clock on Tuesday. I will be here at 4:30 up in lab if you have last minute questions. You can always text me over the weekend with questions. So study hard. If we have lab tonight, I'll see you up there in 10 minutes.